So teachers, now next very important concept anyway for now also, the board exam, we need to prepare the children very well this particular topic. Mostly we can expect a question from this particular topic, that is a polymerase chain reaction. Most of the question paper when we take, it is a very important question. It has been a very important question. Students generally they write very well only, there won't be big issue, but we need to make sure. Yes ma'am. Yes ma'am.
No, nothing, sir. Nothing, sir.
Okay. So main mistake where they do is they forget to mention the name tag polymerase. Why we use tag polymerase? Teachers, kindly the student need to understand what is the difference between the polymerase and tag polymerase. Of course we know that, but children to their level we need to make them very clearly understand what would happen if the normal polymerase is used for the polymerization. Then what will happen? And what speciality does this tag polymerase has in comparison with the regular polymerase? We need to make the children to understand. If that concept, if the children are able to write it correctly, mostly other things, the steps and all, they will be able to write it very well, definitely. But as we all know about, the CBSE will little try to ask question in such a way that what will happen if a person want to manipulate a single standard DNA? What are the steps will be required? Like that, the questions will be little twisted and asked, so that the children, whether they have understood or not, that has to be known. So if it is a single strand of DNA is found, can we say what are the steps would require, teachers? If a single stretch of DNA, if only in a polymerase chain reaction, only a single stretch of DNA was found. Now, what are the steps would involve?
For single strand DNA, sir, you are asking?
Yes, ma'am. Maybe in an accident or somewhere, only one small stretch of DNA was found. That was a single stranded DNA. Okay. Now that has to be manipulated, amplified. For that, what are the steps would be there?
Denaturation would not be there.
Yes, ma'am. That we need. The children must be able to understand. Without denaturation, directly we can enter into that annealing process, and we can go ahead with that. So the student must be able to understand, if it is separation, then immediately the answer should come to their mind is gel electrophoresis. If it is amplification, that immediately the answer must come to their mind is about this polymerase chain reaction. So we need to make in such a way the students keep this in their mind and they are able to correctly right. And tag polymerase, they must not, of course, the temperature and all, it is not mentioned in our text itself. Of course, they know. They may know that 94 degrees Celsius and then 52 degrees, this cycles, they know that. But in an exam point of view, that no need to be mentioned. It's not mandatory. But tag polymerase, they must write at least one answer. And in exam, if they don't know how to write it in the descriptive type, they must at least able to draw the diagram neatly. Diagram is 100% must for either drawing or the questions will be there based on the diagram. So student must be able to correctly identify which are the steps, which is the prime end, definitely that 5 prime, 3 prime, because that the to write it correctly and should be able to mention in that when they are writing the description also, they must be able to write that 3 prime end, they must keep in mind. And little knowledge of the polymerase activity also will be needed, but of course they know that that won't be a big issue if they don't know also. And this kind of we can put an input, what are the things will be taken into that PCR cyclic mission, what are the DNA sample, primers, the nucleotides, tag polymerase, the buffer, and PCR, everything will be added in the PCR tube and it will be put. That student must be able to understand what are all the components are required for PCR that they have to say. And next one is the recombinant host. Now already we have studied about what is competent host. What is conjugation? When we say these words to the children, they will be able to keep that in their mind for a long time, and without forgetting, they will memorize everything. Next, recombinant protein. This question asked in the exam recently. What is the organism the host is called as in which the recombinant protein is produced? Or the gene is the recombinant protein. What is that particular host is called as? So it is called as a heterologous host. The student must be able to write it in the keyword. Then only the for marks point of view, it is very, very important that we should know. Next, the gene product. The students are not at all aware that they are thinking once we do the research, once we find the recombinant protein, that's all, the job is over. They don't know what will happen if the life cycle of the E. coli or any bacteria, it's just 20 minutes. If at all something happens, if the culture is not again made, then what will happen? Everything, whatever the research has been done by the scientist, everything will go in vain. So far, whatever the efforts were made. The student must be definitely in this point, they must be able to understand what is meaning of continuous culture system, how that laboratory, how that from one bacteria from the previous culture, how it is inoculated in the new plate, and how that continuously it is keep on happening. Why it has to be done only in the laboratory, even when it for a large scale production, the continuous culture system must be followed. Otherwise, the whatever the material produced, whatever the gene with a lot of efforts we have made, the scientist made, everything will go in vain. So student must definitely understand here how in the laboratory or in the industry, how that continuous plates are made using this particular recombinant organism. That will be helpful for them to clearly understand what are all the steps are involving in the obtaining a gene product. And this definitely they would know what are the this particular this machines that, like, bioreactors, what are the capacity. If possible, it can even we can take the children to the industrial visit. We can show them the nearby visit how actually these things are happening. And there is some post they have to understand about what is the job of a person who is working in the R&D, research and development. The person will be constantly doing research about what is the optimum temperature required in the temperature or pH or substrate, various salts and oxygen, vitamins, everything. What is the optimum condition required? Because when we go to the industry, everything economic matter, the money matters. So they should spend less investment and should get the best product out of it. So there is a R&D person who will be continuously doing the research about various temperature. Same microorganism at 22 degrees Celsius, it will yield some amount of product. Same microorganism at 24 degrees Celsius, it may yield much more product. At 26 degrees Celsius, it will the product yield will be reduced. So the children must be able to understand what is the minimum, what is the optimum, and what is the maximum, various conditions required for the microorganism. It is not depending on only the recombinant organism, that heterologous host. Apart from that, the temperature, pH, substrate concentration, everything matters in the production of this bioactive molecules or whatever the product which is going to be produced. And this bioreactor, they must know the type. Usually we compare it also. So the student must know about different labels which is there in the textbook. If they know that, that is more than enough, but they must know that. It will help them to learn better. Next, in the parts of bioreactor, as we said, how the pH is controlled, how the foam is broken, how this oxygen is delivered, what is the need of oxygen. We need to put that. Even we can ask some questions like, we can ask an anaerobic bacterium. We can say that yeast for the production of ethanol, will it require oxygen inlet? We need to put the question to the children. Then we're hearing that the children will be able to say, no, we need to expect the answer. Various answers will come from the children. So we need to make them understand how that oxygen, that is a yeast, will do that anaerobic respiration. Then it won't require any oxygen inlet. That students will, it will broader their thinking. It will, believe me, it will helpful them to think the knowledge which is there in the 11th standard also. so that whenever they are writing some competitive exam, NEET exams and all, they will have an idea. But for sure it will help in one or another way. So that must be understood. The next one, downstream processing, a very important question, repeatedly asked in exams many times. As we all know, student must know what exactly is that separation and purification of the product. They must understand that product will not come with just in a pure form after the reaction. There are many unwanted things will be mixed with that. That is a part of substrate or the cell itself. All those things must be filtered and removed. Right? That has to be packed. Packing also, we need to put an idea, light into their mind. We can take an example of Tetra packing. What is the need of Tetra packing in a milk or any milk product, dairy product? What is the need of the? What is the reason? So this and all, when we put, they will understand that all this biotechnology product, after the manufacture, it needs a safety packing and dispatching. If something happens wrong there, what will happen? It may invite some contamination. When the person eats the contaminated food or eats takes the injections or any vaccine or anything which is contaminated, that will be going to, for sure it is going to be life threatening for the person who has taken. So children must understand it is not alone depending on the purification and extraction, and also for a dispatch, packing we have to give equal weightage. Why there is an
Sir, your voice is breaking,
various
sir.
Oh, sorry,
Excuse
ma'am.
me, sir.
Yes, ma'am. Yes, ma'am.
Your
Now am I
voice is
audible, ma'am?
breaking. Now it is audible.
Oh, thank you, ma'am. Thank you. So that dispatching also the student must be able to understand. Otherwise, it will be a that they won't have an idea how the things are happening. That we need to definitely make them understand. Next, that chapter fully over. So downstream process. About various, especially the cloning vector. A very important questions expected in the exam is cloning vector. The next level it will be that polymerase chain reaction, then gel electrophoresis. So we can order them in such a way. And most importantly, this year it is a really, we can expect some question definitely. Only from this the questions will come. Mostly we can believe or we can expect whatever the questions are there in the exam, that will be mostly in and around whatever the topics we are mostly emphasizing the children to learn. Mostly from that only it will be there. But in that question, children must be able to write it correctly, should be able to get the maximum mark for that answer. That is in the children's hand. We need to prepare them accordingly. Next one is the 11-mark chapter. There's two together, 11 marks, biotechnology, principle and process, and application together for 11 marks. Mostly around 8 to 9 marks we can expect from biotechnology, principle and process, and maximum 2 or 3 marks, or even up to 4 marks we can expect from the biotechnology and its application. And teachers, this lesson, especially when you are having such a slow learners in your class and all, make sure this lesson, microbes in human welfare, and last 2 chapters, these 4 chapters, if they study very well, definitely a very decent pass mark will be there. It won't be much less than the passing mark, it will be more than that only, and they will be able to, they will be in a safe zone of passing the exam. So we need to emphasize there. So next one, sometime I can. Yes, ma'am. Yes, ma'am.
I have some doubts, sir.
Yes, ma'am. Please, ma'am.
Actually, we are showing them bioreactor, no, sir.
Yes, ma'am.
We are showing the appearance of bioreactor.
Yes,
right?
ma'am.
Contents of bioreactor, if you have some pictures, can you share, sir? I mean, how they will load the content, what they will load inside.
Yes, we need to show, ma'am. There is no wrong in that. Even videos you can show.
Pictures you can share, and products.
Okay,
once we are loading the content and how it is coming as the products, that pictures I could to find.
Yes, ma'am. Either I will try to find a video or
Yes.
I will try to find that
So
photos.
video
ma'am.
I have seen like it's like lengthy video. I need a correct picture when we load how
Oh, yes.
how they load the content, and finally when they get the product, it will be in which form product.
Definitely, ma'am. I will be sharing that. Yeah, surely.
Sir,
Thank
and
you.
one more doubt, sir.
Yes, ma'am. Yes, ma'am.
DNA, what we are using to construct a recombinant DNA. For example, like we are taking one particular desired gene only we are taking.
Yes,
That
ma'am.
is found inside DNA. So that
Yes, ma'am.
DNA, when we are isolating, we are subjecting to some enzymes no, sir. For
Yes,
example,
ma'am.
when we are taking from bacteria or when we are taking from plant cells or animal cells, we are subjecting that from particular enzyme. So
Yes,
full
ma'am.
length DNA, we are going to take only a particular part of the gene, no, sir.
Yes, yes, ma'am.
But when we restrict DNA, will the
Hmm.
whole DNA will be restricted? I mean, with the help of restriction nuclease.
That will happen when we are using a linear DNA. When we are taking, for example, linear DNA, when we are using the restriction, particular restriction enzyme, maybe we can say EcoR1 we are using, it will make that particular DNA into two different fragments apart from our gene of interest. Total, we need to assume 3 fragments will be there. One is our choice gene. The other 2 fragments will be obtained.
So that
If-
gene of interest we can identify after gel electrophoresis, sir, or how to when we can identify that gene
Exactly, ma'am. Yes, yes. Here only we need to take the help of Human Genome Project. We already have been sequenced the DNA. Say, for example, in a my choice DNA is located in chromosome number 21. My chromosome, my gene of interest will have 5 repetition of guanine or 7 repetition of some nucleotide. I will know already because of the Human Genome Project. All these things are sequenced. So what when identifying a restriction enzyme, we will find it is not just a piece of cake to take any restriction enzyme and use it. It requires a lot of research to understand where exactly this particular gene of interest will be cut and the nucleotide sequence. After that only we will be able to cut that particular DNA. And when we are cutting that, we have the size. Generally, always it will be 0.1 kb to around a 2 MB. That much size that gene will be varying. So when we are cutting that according to the Human Genome Project, we have classified all the genes according to their molecular size, the weight. When gel electrophoresis we are running, the choice DNA, that will also say, for example, in the Human Genome Project, we have recorded our choice of DNA is around 0.2 kb. So when we're running, that there will be scales that and all we are not seeing here. There will be a scales of what is the size of the DNA which is running on the gel electrophoresis. A bigger DNA will be staying back and a smaller DNA will be much farther in that. There, when a scale where up to 0.2 or 0.1 or 0.3, in that range, whatever the DNA is there, that we have to take. We again, we need to keep in mind, we won't take just one piece of DNA. Whatever there in that range, we will take around, if say, for example, 10 DNA fragments are there, all the 10 fragments, we will take it. We will run into the next PCR. We will go do it in the vector. We will be annealing with the, joining with the DNA vector. We will transform. From that, in which DNA we are going to get the positive result in a selectable marker, that only we will be taking for the, that there only we will come to know which DNA was ours. Until then, we won't know which DNA was ours. It is all like a hit and try. There's a trial and error method only. So
Then
10
after
DNA.
that only we can amplify now, sir.
Which one, ma'am?
After that one we can after we find our desired gene only we can amplify.
Yes, but we not that only our desirable DNA will amplify, ma'am. Whatever the DNA in that particular weight range, whatever we are taking, everything we will amplify, ma'am. 10 simultaneous plating will be done. So for students' sake, we are telling that choice DNA only we will be amplifying and will be doing. But we won't know
Yeah.
which one is our choice DNA. It will be in that range only. In between range, it will be there. So we need to run everything. At last, when we are getting the product only, we will come to know which one is our choice DNA. That's why we call this as a very tedious job, having simultaneous plating and all this thing. That's why we call it as so. Thank you, ma'am. Very nice. Mostly teachers, next when we are teaching, mostly we means the students also don't give important weightage to that. We also need to make sure where and all the critical research area is going on currently. So directly it is not depending on one research field. Doesn't mean that finding a choice DNA, amplifying that, or finding and molecular this scissors, and it's not like the entire thing cannot be done by just one scientist. It is not possible. So scientists can play their role, vital role in different areas in biotechnology, not necessarily into the whole segment. Student must be able to understand identifying one catalyst itself is how much big task they must be able to understand. It is not just a very easy thing. So they must understand, if a scientist is able to identify one particular organism or one particular enzyme. So recently one German scientist has discovered a microorganism, a bacteria that can digest plastic. That's all over. One research is over. That itself took more than 7 years, 8 years for the scientist to discover. So it is not possible for him to identify what is the enzyme is there, how it is functioning, how can we do that biotechnology in that and all is not possible. So student must be able to segregate that, either finding one catalyst or one organism, that itself is one research. Then the next research is whatever the organism we have found, or whatever the enzyme we have found, in what optimum condition they are working. We need to find that. What optimum temperature is required, what optimum substrate concentration is required. All these things they have to do the research. That also comes under another research. And last one is devising a proper downstream processing for isolating those products. That also comes under a different research area. So student must be able to understand clearly. It is not simply a thing that one person can discover everything or do everything. So any one also we do. The further step, the student must be able to how that different people work together to achieve a particular things in the field of medicine. They must be able to understand. Next one in agriculture, this and all very we can expect very well in the exam. And here we need to add the value based question. Usually teachers, I always take one WhatsApp message and one photograph with me whenever I'm going to the even physical class. Whenever I'm visiting, I do that. Just I'm sharing how it works out for me. I'm just sharing. In the WhatsApp message, if you see all the time, there is a message like, we should not eat genetically modified organism. It's bad for health. We should not eat agrochemical based crops. The chemicals, fertilizers are spoiling the soil profile. So on. So the lengthy messages will be there against the use of agrochemical based agriculture, this everything. I'm not in support of that, but we need to make the children to understand or realize the situation why agrochemical based agriculture is currently in use. That is much important at the point of time, present time. So you take that the Great Bengal famine, that photograph, you can see the people standing with all skinny, fully the bones will be visible, how they won't be having even food for one day or one time for a day also to eat. We need to show to the children, and we need to ask whether we want to live in such a situation or we want the agrochemical based agriculture. That time when we are all suffering a lot of acute the shortage of food grains, and we were all suffering a great hunger. At the time, this agrochemical based agriculture, Swaminathan's, the plant hybridization technique, all these things only gave us the helping hand to overcome this food grain shortage. And today we are mostly a self-reliant country in terms of all our food produce. This all because of the agrochemical agriculture. We need to use the chemical fertilizer, we need to use the pesticides, and we need to grow the crops. But students will think, why can't we go with the organic agriculture? So we need to make the children to understand why organic fruits are much costlier compared to the conventional agriculture, that is agrochemical based agriculture. We need to ask the question to the children. Of course, in organic agriculture, we don't use the chemical fertilizer. We assume we don't use chemical fertilizer or we don't use the pesticides, but it is produce will be very, very less. Because of that, it cannot fulfill the need of all the growing population. That student must be able to understand. And genetically modified organism is the only hope we have it in the future, not immediately, though very in the market, and India has allowed only around 4 genetically modified organisms only. In all over India world, if you see also, maximum 9 genetically modified organisms are used, not much more than that. This area is completely under research. Only this will put an idea into the mind of children how this in future we can depend on genetically modified organism rather than this organic and agrochemical based agriculture. Student must never find fault with the agrochemical based agriculture. That also we need to make sure, though it involves chemical fertilizers and all, we need to make the children to understand it is not completely, we cannot rule out that we need to make the children to understand. Same way, genetically modified organism, when it comes, maybe next month onwards, even in our all over India, we will be getting this golden rice only. There is a news article there. So children must understand, they must be able to link to their surrounding. Maybe next in a couple of months, whatever the students are going to eat, maybe from the ration shops, PDS and all, will be definitely a vitamin A enriched rice only, golden rice only. It is a possibility. And in what way, here itself we need to link this concept of genetically modified organism, how better it is comparison with the apomixes, how it can be brought into this idea. We need to link the topic into the surrounding areas, then only the children will be able to understand clearly. And these points maybe it can be expected in question also. So we need to make the children to understand that line by line. It's a very easy topic, I believe so. Children will take it like a very easy concepts only. Next one is the pest resistance. What is the students are not even understanding, or they don't know what is the difference between a pest and an insect. They don't understand. First definition we have to put to the children what exactly the insect is and what is the pest is. Generally, we can even use the word in English, don't pester me. This student is pestering me. What is the meaning of that? We can ask the children, and children will say it is called disturbance. So we have to say that a pest is nothing but an insect or anything. It's not necessarily only insect or any living thing that causes economic loss or causes trouble to the farmer is called as pest. And why the pest has to be reduced? When though we call it as a pesticide, we call that chemical as a pesticide, but it is not going to kill the pest alone. It is going to kill even the beneficial organism also. This idea must be there in the mind of the children. All the children must have that particular idea, teachers. Some BT cotton, BT corn, BT rice, all these examples we can give how this has been a very important one. Before better we can inform the children about what are all the troubles faced by this particular farmers in previously when they were using normal cotton, how much economic loss used to happen. We need to link that little idea with their social, that economy we need to little match with that. We need to say that when a huge loss is faced by the farmer because of the normal cotton, and how the cotton is actually destroyed by the various type of pest, that and all we need to tell to the children. Then the students will understand there is definitely need of the improvement in the cotton. Here again, as I always say, we need to put the idea into the mind of the children that here what we are doing is not a social service. Here also we are exploiting only. Here also we are exploiting the cotton crop. We are destroying the original variety, wild variety, and we are making into a biotechnology, and we are making a BT cotton. We are exploiting the cotton for our personal use. That we need to highlight to the children, because there is also a question how human is exploiting all this BT cotton and all. There is a question also is there. Student must be able to answer that. Then they must know what are all the different insects are there that usually target this particular cotton plant alone, and how this particular Bt toxin produced by the protein. The children will be mostly clear only when we are teaching this. They won't have doubt. Why? Because already they have studied this in the previous lesson, that microbes in human welfare. They have already studied that. Here they are going to, there is no technical terms also, except that the protein name they have to memorize and what it controls. That alone they have to keep in mind. Otherwise, all the other things are already there in the, already they have studied in the particular previous lessons, they have understood. So here they have, we need to emphasize on different toxin protein gene and what are the particular type of insects are controlled by this particular gene. We need to make them memorize. There can be a question, many time repeated questions are there on highlighting this particular concept alone. Next area, this alone students will be facing little challenging. I won't say too much, a little challenging. As always I say, it is, they will face challenge because they won't understand, they won't recall the previous knowledge. Try it once and see. When before we start this agriculture, this nematode, we will stop it after the Bt cotton, will stop for a while and we'll pull the idea from that molecular biology. We can ask some questions from the molecular biology. Why RNA is always a single stranded? Why it is not a double stranded one? What is the reason? Why both the strands of DNA do not act as a template to form the RNA? We need to ask the question to the children. And children will be able to understand, yes, that RNA should be a single strand, then only it will be coding for a particular sequence of amino acid. One type of protein only will be produced. If both the strands of RNA is there, either translation will not happen or it will form two different proteins for the same phenotype. So it will go wrong. So it will not happen. RNA should be always single stranded. This answer we need to pull from the children mind. When they say that, then next, the gene. We already have seen different type of gene. I asked you already in the beginning itself. Whenever we are teaching molecular biology, kindly teach different types of genes available. As I already said, housekeeping genes, the special genes are luxurious gene, or this transposons. All these genes, when we are telling to the children, they will be able to link. Now there is a need of the knowledge of transposons are very, very important here. So we can tell the children about this particular nematode, how it lives in the root system of this tobacco plant, and it reduce the yield of the tobacco. So we need to tell the children, if the yield is reduced, what will happen to the farmer? How it will affect our economy? We need to put an idea to the children. Now, novel strategy which is developed by the scientists, which will make the RNA to silent. It will make the silent RNA to keep quiet, and it is called as in RNA interference or RNAi technology, where RNA will be made into double stranded RNA. As a result, the translation would not occur and the plant will no more required by the nematode, and nematode will leave that place since it will become a hostile one. This particular nematode lives in the mainly in the root system of many plants, especially in the tobacco root system also it lives. And here we must understand what is the source for the complementary RNA. Many times asked in the exam, students will be having the doubt. So two sources are there. One is either transposons used directly or RNA viruses also used. Both are used as a complementary RNA for the RNA which is already there. So complementary RNA. Second thing, the vector system students are missing usually. They won't write it. In a hurry, what will happen? They will write RNA inference technology. They will write the source for the complementary RNA. They will write, they will forget how this complementary RNA is delivered into the host. That is a tobacco plant, how it is delivered. That they won't write. So we need to say that using the agrobacterium vector only, it is delivered. That student must be able to, again, has to understand that. So now one students may ask, this is a very important topic also. In a eukaryotic organism teachers, as we already said, the expression of gene is controlled in different levels we have seen. In our body naturally, say for example, if one mRNA is already produced, Now later the body decides there is no need for that mRNA. Some parts of that mRNA no need to be converted into protein. No need of translation. Then it produces some type of RNA in our own body itself. Those RNA will go and bind onto this particular the sequence which need not to be expressed, and those sequence alone will be not translated. Can anyone recall, teachers, which one is that? What is the name of that particular that RNA? You can try to recall. In our body, naturally in eukaryotic organism, those RNA will be produced in order to do that silencing. It naturally happens in our body. But in the nematode, what we are doing specifically to the nematode specific RNA, we cannot find it in the body itself. So we are finding it from the other source like RNA virus or transposon we are finding, and we are silencing that. But naturally in the body itself, sometime when there is no need of that mRNA to be expressed, some RNA will be produced to silence that same procedure. Can anyone recall, teachers? Try to recall. We all will together only going to.
Mobile genetic element transposome.
Yes, ma'am. That is there in the mainly outside.
Only that all around, sir.
Yes.
Complementary means mobile.
Yes, ma'am. It's a siRNA, ma'am. siRNA. We have in the body itself. Mostly this is used for the controlling of the gene expression. Yes. Thank you.
Short interfering RNA.
Yeah, exactly, ma'am. Exactly.
No, I
know,
had a confusion. Is it SN or what?
When we say one word, she will immediately recall. I have understood.
No, sir, actually, I'm having a SNRNA, HNRNA, or SARNA. I was having a doubt.
Okay.
Then
Very
finally,
good. Very nice.
SA, now I just recalled.
Okay, then challenging another one. What will be the number of nucleotide will be in siRNA? Anyone can say.
Number
how many long? How long will it be? How many nucleotides will be there in a siRNA?
Number of nucleotides. I'm not remembering numbers.
It's okay. Around 7, 19, like that, very short only. As already it is said, small interference only. So it will have very short stretch of nucleotide like 7, 9, 19, like that only it will be there.
Okay.
Thank you. So next one is the medicine. So medicine, we can expect both insulin as well as gene therapy. We can expect in the board exam very well. We can expect. And we can train the even not before now we can train the students to make the children to read and write very well this insulin and that. Teachers, a kind request. Nowadays children are becoming very careless about their health. You can see after the lockdown how much their health has been. I mean, it's become very poor health. They don't care about their health, personal health and all. So here there is no wrong in giving a little advice. Usually we can do biology, even if they don't study in after 12th standard. I say always to the children, after 12th you may study any subject, any engineering, anything you can study. By learning biology, you will at least understand about your own health, how to keep your own health. If you are going to a doctor, they are saying something, they are making a blood group, test, and what is there, the blood level and all. What is there about the cell count and all, they must be able to understand when
Yes,
CT scan has to be done. When
give it as a project, sir. 11th standard, whenever I'm
Yes,
taking
ma'am.
body fluids and circulation, I'll ask him to take a blood report. In that,
Very
I'll ask
good,
him to
ma'am.
note this WBC, monocyte, lymphocyte,
Yes,
basophils, eosinophils. I'll ask them to note.
Excellent, ma'am. Excellent, ma'am. Yes. Very nice, ma'am. Very happy to hear that. Very nice. So yes, teachers. No, in that same way as ma'am said, or any we can all use this knowledge of the children. Say for example, nowadays India #1 is not in infectious disease. We are all #1 in the non-infectious disease, non-infectious everything, coronary artery disease, many non-infectious disease which is related to our lifestyle. So student must be little made them understood what is the difference between different types of diabetes. Type 1 diabetes means what, type 2 diabetes means what. Little knowledge we have to throw into their mind. so that they will feel their health conscious also. They will develop instead of just studying, writing in the exam, getting marks and forgetting. They must use it in their personal regular life also. They must be able to use it. Then this insulin, mainly what we are going to give in the ready-made insulin form, it is only on adult onset of diabetes only we are going to give it, because there only the insulin will not be produced. In there are type 1, type 2. In the type 1, the insulin will not be produced. In the type 2, the insulin will be produced, but it will not be uptaken by the cells to convert that excess glucose into glycogen. That will not happen. So there are two things student must be able to understand clearly. And second thing, why nowadays people are again asking why there are number of more and more number of diabetes patients are increasing. Actually, it is not increasing. It is all as it is the number only. But previously, it was not easy to treat that insulin. Why? Because whenever they are having this diabetes, the only source of insulin injection will be either from the slaughtered cattle and pigs. We have to take the pancreas, we have to squeeze it, we have to take the insulin. As much as possible, we have to purify, and then we have to inject it to that. And then it is a problem associated with that. Quantity also very less will be there. More and more people cannot be given this insulin. Second thing, those who are given insulin also, they will develop some kind of allergic and other type of allergic reactions will be there, because it is definitely foreign protein. Recently also what happened, transgenic pig, from the pig, the heart was transplanted to the person. The surgery was successful. It was a very nice surgery. It was excellent. But unfortunately, what happened? The person has died because though it is from the pig, transgenic pig, it is transplanted. It is a foreign organ only. The immunity of that person did not accept it. As a result, in a very few within 2 weeks or 3 weeks, the person has died because of that cardiac arrest or the heart attack. The person has died. So it is definitely a foreign only. It is not native to the body. And how this can be made? So we need to put the challenge to the children. Because already having studied about biotechnology, they must be able to draw a flow chart of a recombinant DNA technology for this particular insulin. No need to directly straight away go and explain them about A chain, B chain, C chain, what is prohormone, what is a hormone and all. No need to explain immediately. We'll give one chance for the children to draw that flow chart of how to make that artificial, that recombinant insulin, how it can be made. All they have to do is they have to assume the choice DNA is the DNA that is going to code for insulin. That's all they have to keep in mind. And later we will reveal to the children what problem is there because of doing so. Whenever we are isolating such a gene and we are doing this rDNA technology using such a gene, what a problem we are facing, the scientists were facing in getting a hormone from the prohormone. Then we need to explain to the children the A chain, B chain, all this thing. If we need to go little extra, we can say the number of number of nucleotides are there, everything we can explain. But I am mostly focusing on the textbook, because 12th standard student, they have to prepare only for the mainly board exam. They have to prepare. Otherwise, what will happen? They will write all the extra points and all. They will write it in the exam. They will forget the very important keyword that they must write it, that they will forget. As a result, it will lead to the reduction in the marks. That's why mostly will be emphasizing mainly on the what is the textbook concept. But there is no knowledge wrong in giving little more information to them, so that their idea will be broader about everything. So pro hormone, what was the challenge? Later, we need to explain to the children how this American company who has made this Humulin, how they are success in obtaining this directly the matured hormone without even getting the pro-insulin, how they were successful in that, how the disulfide bridges were made. All these things we need to explain to the children. Then student will be able to keep in the mind for a long time. All I request is let them come with an idea. We'll put the problem in front of them. Let them come with the different ideas. Then we can reveal what is the correct answer for that particular what could be, we cannot say exactly this is the answer. We can say what could be the most appropriate answer for this. And here also we can link the 11th standard concept a little, we can touch. What will happen if insulin is given in a oral form? Why it is insulin is always made in the injection form only? Why not in the oral form? We can ask. Yes, teachers, why not can be made into a tablet form? Why only always injection?
It will be digested.
Yes, ma'am. Very good. Shanmuga Priya, ma'am. Now only open the mouth and said the answer. Thank you, ma'am. Yes, as ma'am said correctly, it's a protein. The insulin is nothing but it's a proteinaceous one. When it is consumed orally, you know, very important enzyme that it has to pass through in our stomach. It will very easily get digested into different amino acid, so that it will be definitely a waste. The insulin cannot serve its purpose, so it has to be given in the injection form only. We need to link the concept of 11th standard little. We need to touch it here. Then gene therapy again. Here gene therapy, student must understand what are all the genetic disorders are there. And here itself, student also must understand all this gene therapy are under consideration only. It is not confirmed. It is not therapy is not readily available for all the disorders. The research are being conducted by various scientists. So they also can do research in future if they want. There is a very bright future is available in the field of gene therapy. We have to tell to the children. And here gene therapy, what exactly the gene therapy? What before we start gene therapy, they must know at least what are the genetic defects are there. So they have studied more than 10 different types of genetic disorders right from the beginning of the 12th standard. So we have to ask them to recall all the different type of genetic disorder. That too in the upcoming academic year, that will help them to prepare for the year-end exam. Because term 1, term 2 together it is going to be there. So it will be really helpful. We need to ask question from the principles of inheritance and variation. What are all the disease genetic disorder they have learned? In the molecular biology, what are the names they have learned? We have to make them simply recall. Then we have to say one more disorder is there, that is called adenosine deaminase deficiency. In the book, they have not given much about the enzyme. They have simply given that it is important for the immune system functioning. We need to little elaborate that how it is very useful for immune system. Since already they have studied about lymphocytes and all, what is the role of this particular enzyme in the lymphocytes? All the thing little, not too much, at least 1 or 2 words, 1 or 2 sentence, we have to explain to the children. They will be able to understand. And what were the treatment method available for the person who has this ADA deficiency previously? There were only 2 diseases were there. 2 medicines were available. One is bone marrow transplantation, enzyme replacement therapy. So what is bone marrow transplantation? Again, we have to link it with the graft rejection immunity. What will happen if from a non-compatible donor, if transplantation happen, what will happen? From the compatible donor, if it transplanted, how it will be accepted? We need to say that which immunosuppressive drug will be given to the patient when we are doing transplantation. This and all teachers, like linking biology is more or less like a flexible lesson only. A little we are, by recalling the content from previous lesson and forthcoming lesson, we will be able to make that this topic also in the mind of children very well, and the previous topic also can be put into the recalled very quickly, or it can be recalled. As a result, student will be knowing the content to the maximum level. Of course, after the classes, we are going to revise once again, that and all is there. But this kind of linking with the previous knowledge, previous chapter, not required to 11th standard, at least to the 12th standard, what they have studied, we have to little touch that lesson and we need to come back. So what happens in enzyme replacement therapy? Why this both the treatments are not completely curative? What is the need? We have to make them understand. Then gene therapy, though gene therapy can give a permanent cure for this particular it is not completely curative. The children must be able to understand what is the stem cells. Nowadays, if you see in the advertisement and all, stem cells banks are very common. Now can store their child's stem cells. When the child grows up, if the child has any problem, that stem cells can be used for curing that particular child's whatever the disorder the child may be having in the future. So then we need to give an idea about stem cells here. How the stem cells, before starting gene therapy, the stem cells idea we have to give. That stem cells is not available in the 4-year-old girl. That student must be able to understand. Then we need to make the children to understand how in the 4-year-old old girl the gene therapy is done, how for the embryonic stage, during the embryonic stage of the baby, how it can be done on the stem cells. Both they have to simultaneously distinguish and they have to learn. Then only they will be able to keep in mind for both the topics. They will be able to keep in mind. Or we can even ask questions like, there are two patients. One is patient A who is having 4-year-old baby who want to take treatment for gene therapy for ADA deficiency. The other one is the mother came to know through the genetic testing that in the embryonic stage it has to be performed. Who will get the permanent cure and why? We can ask questions. Same way, what is cDNA and how this cDNA is transferred into the host, that is lymphocyte, how it is done. The retroviral vector, they have to mention the teachers. Next one is molecular diagnosis. Already they know PCR and all they studied, but what mistake the student, some of the students are doing is that whenever the PCR is asked from this particular lesson, they will be writing about what they have learned in the previous lesson. Or if they are asking about the previous lesson, they will be writing about what they are studying here. So we need to give a clear idea whether the question is about molecular diagnosis or is the question is about that PCR technologies, the steps involved in PCR or amplification. So student must be able to distinguish between these two and they must be able to understand. And here we need to highlight what is the drawback or limitation of the conventional method of diagnosis. Say for example, one person has disease. If we are going for a blood test
Take a
and
long
PCR.
time to detect, sir.
Yes.
It take a lot of time for detection itself.
Very
Whereas
good,
in PCR,
excellent.
a short time, a large number of sample can be detected. So that,
Excellent,
PCR
ma'am. Excellent, excellent, ma'am. Very nice. Yes, definitely yes. This is what we are expecting. So the conventional method, as ma'am also added, so that blood. By the time we do the blood test, we come to know about the disease. The disease is already in the advanced stage. Urine test, blood test, and all will be only done after the appearance of symptoms. Once we get the symptom, we go to the doctor. Then doctor will recommend for a blood test or a urine test. All this test will be done only after the appearance of symptoms. Am I right, ma'am?
Yes, sir. Yes, sir.
Yes, ma'am. So on the other hand, this RT-PCR or PCR, ELISA and all can be performed much advanced, even very long before the appearance of the symptom. I always ask the question to the children teachers. A person has got COVID-19 positive. The person has developed the various symptoms. Now the person goes to check PCR, RT-PCR for checking the presence of COVID-19. Is it recommended or not? I asked the question to the children. What will be the answer, teachers? If you were in that situation, I am a student, I am asking such a question. Yes, ma'am. Shanmuga Priya, ma'am, want to say something?
Sir, yes, it is recommended with RT-PCR.
Okay, ma'am. Good, ma'am. Amada, ma'am, what do you like to say?
Yes, sir.
Yes, ma'am. Is it recommended, ma'am? One person got symptoms. He goes to the doctor.
Yes, sir. It is sure. Sure, he has to undergo PTA, RT-PCR test.
Okay, ma'am. Okay. Actually, teachers, sorry to say that, actually, it is not recommended. Why? Because already the symptom has appeared. then there is no point in doing the PCR.
Then
Why the
why
RT?
hospital they are doing that, sir?
Yes, ma'am. Yes, ma'am. Why they were doing that? Say, for example, if I got COVID-19, I have spoken to some of the people. I was in communication with some of the people, contact with some of the people. We don't know whether they got COVID or not. Symptom has not yet appeared. So what I have to do, RT-PCR has to be done to those people to find whether they are having COVID-19 or not. If they are having positive, they have to be isolated so that it will not spread to other people. Only for the screening purpose only we can use RT-PCR, not after once the person got symptom. What is the need of RT-PCR? Already we know that PCR is the person is having COVID-19. With the symptoms, we can diagnose the disease. No need of PCR. PCR will be performed only when we don't know whether the person is having, the person is exposed to the virus or not. Until the symptoms is appeared, at the time only we can do PCR, ma'am. Do you agree with me, ma'am?
Yes, sir. Yes, sir.
Thank you, ma'am.
Yes,
Thank you.
yes.
So RT-PCR, we can put this also to the children to make them understand. So next, the PCR, what are the steps are there? We have little images and all if we show, student will, otherwise we cannot bring the PCR machine and we cannot show to the children teachers. So some pictures, even ask them to make it in a chart and make it in a school classroom, it can be displayed so that they will know about how this PCR machine, a small machine, it can be done. All the performance of the test, diagnostic test can be done. The student must be able to understand the PCR tube, how
But
it can
in
be done in the
NCERT books, nothing is given there, sir, for molecular
Yes, ma'am.
diagnostic. But whereas
Yes,
in
ma'am.
the state board book, see, sir, how it is given. See, denaturation, every step is given. Is it visible for you, sir?
The students, ma'am. No, ma'am, not now. Yes.
I don't know how to
Uh,
show it. This is the book.
Now
See,
you
sir,
can show,
these
ma'am.
are
Now only you are seeing. Yes, yes.
denaturation, annealing, extension. Each
Yes,
step
yes.
they have given us a pictures are in state board book. It is given, sir.
Yes, ma'am. That's will be there for the teachers only. But state board, what will happen if one paragraph we are studying, the student will be able to understand on their own. But when they are reading NCERT book, it is not so. In one paragraph, when they are reading, there are hundreds of meaning will be there. Am I right, ma'am?
Yes, yes, sir.
Yes, that we only can put them. That's
But
why
picture
it's a,
can be given now, sir. Picture can be for students' understanding purpose.
yes,
Picture
yes.
can
They
be
will
given.
they will be revising, ma'am. Very soon they will be revising the book. Maybe we will expect in the next print. Maybe it will be there or not. We will expect, ma'am. Even if you see that a cell cycle was given wrong in the old NCERT book, and
So-
new it is given different. Now what happened? Many students are having still the old book only. Actually, the diagram is a wrong diagram. So as a result, what happened? We have to consider writing both the diagram, whatever they are drawing, we have to accept. Same with tRNA also, if you see, old book, it will be a different diagram, a new book, it will be a different diagram.
Yes,
We
sir.
have no option. We cannot force all the children to get only the new book. We cannot
New book,
force.
yes, sir. That one we
Yes.
cannot insist, sir.
That's why what happened, we have to consider, even if it is a old book diagram, sometime we need to
Let them
accept.
follow.
Otherwise, yes, ma'am. Yes, yes. Thank you. Yes, ma'am. So next, this especially for the cancer patient, cancer will become much advanced stage when it is showing the symptom. So in that case, what will happen? It will become untreatable. Student must be able to understand, teachers, that cancer will become untreatable. As a result, if the cancer in the, say for example, in family somebody is having cancer in the history, so we can detect early, or on a periodic basis people can go to the doctor and they can check for the presence of cancer. So early if we detect, we find to come to know about the particular cancer presence, we can treat the cancer in a very easily. Same way, various genetic disorder, especially the notorious disease like HIV and all, has to be identified as earlier as possible so that it can be prevented. The student must understand the value behind that. It's a marvelous discovery made in the human history in the field of biology, that knowing the disease as earlier as possible will help in overcoming the serious effect in the future. The serious impact will not be there in the future. Student must be always able to appreciate the teachers. That is very, very important. Same way this ELISA, we can even show the, yes ma'am, Gayatri ma'am, network issue it seems.
Sir, I could not able to.
Join ma'am.
Yes,
At
sir.
least you could have, no, I would have invited you. It's okay, no issues. Thank you. So next one is the ELISA. In that, what
Yes,
happens
sir.
that the student must be able to know how this test is performed to find this antibody antigen reaction. They are not knowing that, ma'am. That agglutination they don't know. They are not using the terminologies. They are not properly using. They are only expanding that this ELISA abbreviation. They are expanding, and they are writing only the antibodies antigen. It is being detected. That's all they are writing. They must be able to write what are antigens here, different type of antigens like proteins or glycoproteins. They must be able to find it. What is that agglutination? They must be able to find. They must be able to know at least. And how this reaction will be shown positive. In negative means what result will be there. Positive means what result will be there. That they must be able to understand. Of course, in exam they are not going to ask that, but they must know at least a little. And next, very important, we can expect this time questions, that is transgenic animals. That what is transgenic animal? Definition they must know very, very well. In exam, they are simply asking definition, and they are asking about mainly this biological products. That is very importantly asked in many question papers. If we can take, it is mainly asked. So we can show some of the transgenic animal. If possible, we can take the children to any veterinary hospital nearby to show how the laboratory animals are looking, how they are put it in the laboratory, how they are handled, like how they are exploited. We can say that why here we can even say about Congaium cow, why it is a serious cause of concern that we have to protect that particular breed of cow. Or various dogs available in India, like Tamil Nadu, Rajapalayam dog and Sipipare dog and all. We are saying why it is a cause of concern when the number of those animals going very low. Why it becomes a cause of concern? We human being, we are all exploiting it. We want to go with the hybrid varieties like Labradors, that what is a German Shepherd, like different dog we need to go. Same way cow we are saying means we have to go with this Swiss cow, the like different hybrid
Jessie.
cows only we are going. Jersey, yes ma'am, very nice. And we are ignoring the wild varieties. As a result, the wild varieties population is gradually decreasing, and one day they may become extinct also. You may find very well this donkey. Last I have seen around 15 years or 20 years back, I remember I have seen the donkey in the street. Now, after this many years, I am not able to see even just one donkey also. I wanted to show to my kids that how a donkey will look like. A real donkey I want to show. I am not able to show it. The sparrow I want to show to the children, it is very rare to find. They are not.
Not able to find the pig.
Yes, ma'am.
Nowadays, pigs are also not.
Exactly, ma'am. Yes, many animals which we have seen as a small children, now we are not able to see those animals. That our next generation, they are not able to enjoy that the biodiversity. They are not able to see that. So we need to put a word here to the children how this is actually a kind of exploitation. We need to put it to the children. In exam point of view, mainly teaches this particular question, that insulin like growth factor, to study the normal physiology and development. The insulin like growth factor is very importantly asked in the exam. And next one is the human alpha 1 antitrypsin, and also the human alpha lactalbumin. These two things children must be able to correctly answer, teachers. The human protein like alpha 1 antitrypsin, which is used for emphysema. Here we need to say what is emphysema. We need to have read in very long back, I think so they would have forgotten. We need to make the children to recall. Yes, teachers, what is that
Lung
emphysema?
Lung
Somebody
disorder.
wanted.
Lung disorder.
Lung
Yes,
disorder.
ma'am. That actually, that particular, yes, ma'am. Alveoli will get damaged. As a result, the surface area for the diffusion will be decreased. The people will not get enough oxygen. Can anyone say what is a major cause for emphysema, teachers?
Smoking.
Yes, ma'am, exactly.
Tobacco smoking, sir.
Yes, smoking. Yes, ma'am, smoking, mainly smoking. So we need to tell to the children that smoking has a serious impact on the lungs. To treat the disease, we have this alpha-1 antitrypsin, which cannot be produced in the human body. It has to be produced with the help of a transgenic animal only. So this is not only treating emphysema, and various other proteins are produced using transgenic animal to treat various disorder. You know, when a person get the phenylketonuria, the person will be mostly like, you know, more or less like the person will die very soon. When it's a genetic disorder the person is having, it will lead to accumulation of that phenylketonuria. Ketone bodies will be more in the urine and the person will mostly die. When a person gets cystic fibrosis, the person will not be even able to breathe properly. When a person born with cystic fibrosis, the person will definitely dead only. So nowadays we are having medicines to treat such a deadly disorder. And same way nowadays the milk, the nothing can equal to the human, the mother's milk we call. But now scientists are trying to make a human same equal to the human protein enriched milk. They are using it rosy cow. They are trying to produce in the human alpha lactalbumin. This we can say. Can you say any goat name, teacher? The transgenic goat used for making the fur. It will somewhat rhyme with rosy. It will start with T. T. It will start with a T, I think. Yes. Rosy is the cow. Tracy is the goat. Tracy is the goat that is used for same transgenic purpose. Second thing, vaccine safety. We can link with the present day situation. Why that Covaxin did not get the approval immediately? Why it was taking a lot of time to get the approval? What is that safety testing means what? Why it has to be tested before giving it to the human being? Why it has to be tested? All these questions we have to put it to the mind of children so that children will be able to understand how we are taking help from different transgenic animal before that is being delivered into the human purpose, how we are using it, especially for the polio vaccine it can be, or COVID vaccine it can be, or any vaccine. The animals are mainly used for, transgenic animals are used for testing, you must know. And when they are writing about a toxicity testing, they must definitely make sure they are writing in the exam also, the transgenic animal carry a gene which make them more sensitive to the toxic substance. Even a little amount of toxic substance are there, it will make it reaction will be produced into the body of the particular transgenic animal. Please show the transgenic animal photograph to the children after testing. When it goes negative, then toxin, it will sometime it will make the people to even cry. That much of testing has been done on the animal. When the allergic reaction happens, you can see a lot of swellings on the body of the insects. They go blind actually sometime when some insects, some chemicals are there. The transgenic animal goes blind. They have become infertile. Various impact is there on the body of those transgenic animal when this kind of test is being done. Of course, they cannot write all this thing on the exam because exam is mostly positive point of view only they have to write, but they must know that what are all impact it has on the animals, how it can be justified. We need to put how can it be justified, we need to ask. Next, teacher, maybe we can expect that GEAC, Genetic Engineering Approval Committee, whenever they are writing, they must definitely write validate and safety. These two keywords definitely they must write in the exam. These two are the keywords. So checking the validity of the genetically modified organism research and the safety introduction of this particular organism into the public use. Because of this only, in India, we have hardly very few genetically modified organisms. We don't have too many. All over the world also we see we have very less number of genetically modified organism. Why? Because we have to address various validity, safety. What will happen if it is introduced? Will it become a threat to any other organism or what? No, not. Few things we can do in the laboratory, but many things are there that we cannot. The immediate result only we will know in the laboratory. The later result we will not get it in the laboratory. For that, we need to wait for even one generation to see what is the impact of that particular in the environment. Say, for example, plastic. When the plastic was introduced very long back, it was well and good. Even till date, our life is unimaginable without the use of plastic. If it is raining also, we take the plastics for any polythenes for the using of some safety point of view, putting the mobile phone or money, something we are using. But after this many years, today only we are able to understand the impact of that polythene on the environment. So there are many things in a when the same thing applicable to student must understand. Same thing applicable to genetic engineering approval committee also. When it is with a blindly, if it is introduced something to the environment, what impact will be there, that will not be understood immediately. It will take a lot of time to understand. Next, biopiracy. Not only basmati rice we have to say, even turmeric, neem. You can take the example of recently issue with Maharashtra farmers with the PepsiCo company. What happened? One type of potato which we are eating in the chips and all, that packaged chips and all, that particular company has sued the case on the farmers because the farmers were trying to grow that particular type of potato in their agriculture field. So the company has actually, the farmers had to face that legal action. But actually that potato was native to India only. Now they have got the copyright, how we don't know. Now, but court has given a proper judgment. The verdict came in favor of farmers only. They recently happened, around 3 or 4 months before it has happened. So those recent ideas and all we need to share with the students.
Even
to make them
Anand Mohan
understand.
Chakravarti. Anand Mohan Chakravarti.
Exactly, ma'am. Very nice. I'm really happy to hear such a kind of example. Yes, ma'am. Thank you. Thank you so much, ma'am. Mamata, ma'am. Yes. So such an example, basmati rice, why it is a big cause of concern. Rights for that particular, and India is being such a generous country. Even for the COVID-19, they didn't get the patent. You know, COVID vaccine, if they taken the patent, nobody would have been able to sell that particular vaccine without India's permission. India didn't accept anything. It has just given the vaccine for free of cost and without any patent it has given. Let all the people use the knowledge. Let all the people be saved from the particular. Yes. But not all the countries are same. They are all making use the basmati rice they have crossed and they got the patent for that. Turmeric they have got patent. Neem they have got patent. Turmeric, neem and all we have been using for ages. Ever we don't remember. But that also they are trying to get the patent. That and all we need to make the children to understand. Then only it's not about forgetting, writing the exam and forgetting. They must know what is the resource available in their local traditional knowledge. It is there. They must be able to understand. If somebody is stealing something from us, the student must know that whether that is belonging to them. They don't know most of the cases. That's why they leave the people to take it away from them. That we need to make the children to understand. And in India is taking various initiative to form this bill was recently passed in the parliament also, and second amendment was also passed in the our parliament. So such a related to patents and all, it's being very viewed very seriously, and people whoever is doing any malpractice of stealing of the product, they will definitely face the law and order issue they have to face. So that we need to put to the children teachers.